Berries have been found to have a rich variety of antioxidants. Antioxidants such as flavonoids help protect our body against reactive oxygen species and reactive nitrogen species. These reactive species have been linked to chronic diseases such as cancer, heart disease, and even neurodegenerative diseases like Alzheimer's. Each of these reactive species contain a radical, which is a free electron that causes damage to biological systems at the molecular level. Antioxidants are responsible for neutralizing this free electron incorporated in the reactive species structure. To determine the antioxidant potential in berries, UPLC or ultra performance liquid chromatography can be used to investigate flavonoid content in berries. In addition to UPLC, a colorimetric assay known as the DPPH assay can be utilized to analyze the scavenging ability of a sample. A DPPH assay is an accepted mechanism for screening antioxidant activity in a sample. Furthermore, scavenging ability refers to the ability of a sample to mop up free radicals. These two tests go hand in hand, as the greater the antioxidant and flavonoid content, the greater the scavenging ability of a sample should be. Moreover, Keiko-2 cells are widely used as a model for the intestinal epithelial barrier. This means Keiko-2 cells can be used to recreate the intestines in vivo. Treating Keiko-2 cells with berries and then subjecting them to a hydrogen peroxide challenge can help induce oxidative stress, which can be subdued by antioxidants. A colorimetric assay known as the MTT assay can be performed following a hydrogen peroxide challenge to determine if berries can help protect Keiko-2 cells from oxidative stress. In this experiment, berries were homogenized and then subjected to an enzymatic digestion to simulate our digestive system. The berries chosen for this experiment were blueberries and strawberries. Samples were taken prior, during, and after the enzymatic digestion to analyze how the flavonoid content and scavenging ability of berries changed as they progressed through the digestive system. These samples were subjected to UPLC analysis and a DPPH assay to examine these variables. In addition to UPLC analysis and the DPPH assay, Keiko-2 cells were grown for 14 days in a T75 flask and then passaged into 24-well plates, which were given 23 days extra to differentiate. These cells were treated with non-digested and digested berry homogenates before being challenged with hydrogen peroxide and then analyzed with an MTT assay to determine cell viability. The UPLC analysis showed a progressive decline in flavonoid content and scavenging ability of the berry homogenates as they progressed through the simulated enzymatic digestion. The flavonoid profile of the berry homogenates was highly varied prior to digestion, and as the berry homogenates progressed through the digestion, the amount of peaks that were not retained was substantial. The DPPH assay demonstrated both declines in the homogenates' free radical scavenging ability and the exorbic acid equivalence. The MTT assay showed significant difference in cell viability of both the pre-digested berry homogenates as well as the post-digested strawberry homogenate in proportion to the hydrogen peroxide treated Keiko-2 cells. The post-digested blueberry homogenate did show an increase in cell viability in proportion to the hydrogen peroxide treated cells, but it was not significant. It was found that the non-digested berry homogenates had a higher cell viability than digested berry homogenates. These findings are further supported by the UPLC analysis and DPPH findings. Overall, this experiment demonstrated that flavonoid content is diminished as food sources like berries progress through our digestive system. The digestive system has huge changes in pH, which can cause degradations of molecules such as flavonoids. The degradation of antioxidants like flavonoids is apparent as shown by the UPLC and DPPH assay findings, which attributes to why there was a decrease in cell viability between non-digested and digested berry homogenates. Even though there was a decrease in cell viability between the non-digested and digested berry homogenates, the digested berry homogenates were still able to provide protection from the induced oxidative stress. Berries have antioxidant potential and are a great source for them, as demonstrated by the initial flavonoid content of the non-digested berry homogenates. However, it is important to be mindful that just because there is a high antioxidant content does not mean that 100% of the initial antioxidant content will be available for your body to absorb.